for me, it's really great to finally meet you, because when I first started getting interested in CCSVI, I thought clearly the action's not only going to be about the neck, right? I'm a neurologist. I don't really care about the neck. Right? We care about the brain.
Yeah.
And I came across your paper, in particular the paper on perfusion.
Okay.
And to me, it's just sort of where the action is. But I think my interventional radiology colleagues, they think the action's out here in the neck.
Yeah.
So here's what I think, and I have a feeling I may be wrong. To me, perfusion, at least the way I do it, which is arterial spin labeling perfusion, is really measuring the movement of water through parenchyma.
Yeah.
But everyone talks about perfusion as if it's the movement of blood through blood vessels. So
Yes.
those are different.
Yes.
So what is the right answer?
Yeah. So I think first of all, let's talk about the perfusion in general. So
what's the definition of perfusion? A general definition of perfusion.
The general definition of perfusion is the blood perfused to the tissue. That's it as the capillary level. It's not the kind of the perfusion through the internal carotid artery. So that's called the blood flow, right? So the perfusion means, as you mentioned, is arterial spin labeling is exactly measure the perfusion from the blood, from the vessel to the tissue at the small the capillary levels. So the whole the CCSVI the concept is interesting because we know that MS is a microvascular disease. We saw a lot of
venular disease.
Yeah, venular disease, and it's small venular disease.
Yeah.
But the CCSVI is talking about is like big veins, jugular veins, the biggest vein, so they drain the brain.
Right.
So it's interesting. It's not kind of like pay attention before. I don't know if it's still. So it's still on kind of work in progress.
The dynamic susceptibility contrast perfusion, which is what you do, is that right?
Yes.
That's using gadolinium.
Yeah.
So gadolinium, by definition, is staying in the vessel.
Stay in the vessel.
So that would sound like it really is blood flow.
That's kind of blood flow.
So it's really not perfusion by my definition.
Not really perfusion, I think. Yeah. So they measure the called cerebral blood flow (CBF). They measure the cerebral blood volume (CBV).
Yeah.
So
I like the arterial spin labeling, partly because I don't have to give gadolinium, so I can do it every day if I want.
Yeah.
Right? I can do it over and over again. But the other thing I like is that it seems to me to be tapping into what we really want to know, which is what's happening to the oligodendrocytes, right? So if CCSV is happening, somehow or other, it's having a bad effect on the perivenular structures, in particular on the oligodendrocytes. So one model is that there's a debate in MS about whether traditional neurologists, they believe the immune system is attacking myelin, but actually no one's ever demonstrated a direct attack on the myelin. There's no antigenicity to the myelin. And then there's the top histology people in the field, Prineus and Barnett, who show the oligodendrocytes are dying first, and the macrophages are coming in later to clean up the mess. So in a way, the question becomes, why are the oligodendrocytes dying? And so to me, the obvious answer is they're in a stagnant environment. They're in an environment where there's poor perfusion. And I think it could be a buildup of waste products. But everybody seems to also equate perfusion with oxygen. To me, those are not synonyms. Whether or not the oxygen level is normal is not a synonym for whether or not perfusion is normal. Am I incorrect about that?
Actually, they are related because there's a flow. The flow brings the oxygen into the tissue. So if there is no flow, there's no oxygen. They are kind of related. But as you mentioned, there's a paper about the oligodendrocyte, the dying. So I think they call it kind of like a type 3 lesions. It's because of the hypoxia. They kind of like related to the hypoxic injury in that type of tissue death. So that's kind of related to the topic today I gave is, the oxygen, there is oxygen delivery problem, and also there is oxygen the consumption by the mitochondria. Mitochondria, if they cannot efficiently uptake the oxygen, there must be a dysfunction of the mitochondria. Yeah. We know the mitochondria produce the ATP, produce the energy for the brain. So if there is a problem of the mitochondria, there should be a problem of the function.
I'm going to say the word mitochondria, because you say it very quickly. So mitochondria.
Yeah,
the most exciting thing you showed, I think, to the audience was that the oxygen concentration was higher, higher than you expected. And I think that's partly because people assume that if there's poor perfusion, by definition there has to be low oxygenation. But you showed that's not correct. You showed that one would predict if you had slow flow, you'd have lower oxygenation. But in fact, you didn't show that, right?
Yeah. So we didn't find the lower oxygenation level in the venous blood. Actually, we found increased oxygen level in the venous blood. That's because I think there's a diffuse mitochondrial problems. Why? There is kind of like the cytokines produced by the vascular inflammation in MS, because we know MS is inflammatory disease, and a lot of repetitive inflammation events occurs. Actually, probably the subtle inflammation events occur every day, but we cannot detect. Probably the patients cannot realize either. So they generate a lot of the nitric oxide, and this nitric oxide is a potent vasodilator. Not only that, it also can inhibit the mitochondrial uptake of the oxygen. So even there's oxygen around the mitochondria, but it cannot use this oxygen. So that's why there's leftover the increased oxygen level in the venous side.
Can we measure nitric oxide using MRI, spectroscopy perhaps?
Mitochondria produce the NA also, but we cannot. There's a lot of things because we have several studies, but on the NA, but we cannot find a strong correlation between NA and the tissue injury. So we cannot directly measure the mitochondrial function, but the technique today I showed is we measure the venous blood oxygenation level. That's kind of indirect, the indicator of the mitochondrial function, I think. So there's another story is the oxygen delivery, because we think this overproduced the nitric oxide, if it persists for a long time, EMS is the case, and can desensitize the vascular smooth muscle. So the vascular smooth muscle, once desensitized, it cannot send the oxygen efficiently to the neuron, because we know once the neuron, we perform some task, the neuron is firing, then they draw the blood immediately to supply the oxygen, for the neuronal cells use. But if there is a problem of this vascular, we call it vascular reactivity. Basically the smooth muscle can contract, send quickly or dilate to the site of the neuronal activity. So when this mechanism is disrupted, so the neuronal cell active but cannot draw the blood immediately. So there's no oxygen sent for them to use. So it's going to cause, we think it's caused the activity-induced hypoxia. So once this hypoxia for a long time, then as the patient's brain is going to be dysfunctional.
How are you going to settle the issue of whether the mitochondria is in fact at fault for the elevated oxygen you see in the venous blood?
How are you
going to answer that question?
How do we solve that?
Yeah.
This is a little bit difficult, to really pinpoint. The oxygen level increase is because because of the mitochondria only dysfunction. Because we know that if the neuronal tissue die, neuronal cell, we know there's an atrophy, there's neural degeneration. If they die, the cells are died, so they cannot use any oxygen, right? So compared to the controls, in general, MS patients probably have a higher oxygenation level in the venous side left over. Yeah. But how do we differentiate them? That's a little bit difficult right now. But we found, interestingly, there's like 20 to 30% decrease of oxygen consumption in MS patient group compared to there's only 2% tissue loss, brain atrophy in this group. Yeah. So you cannot explain, the 20% loss of the decrease of this, the oxygen consumption based on only the 2% of tissue loss. There must be something else beyond the tissue loss. So we think that's because of the mitochondrial diffuse dysfunction.
PET scan or SPECT-CAN or spectroscopy, none of these methods can look at nitric oxide directly?
No.
Because it's such a short-acting molecule?
Very short, 10 seconds, so just the last. Yeah.
That was a little more technical probably than you were hoping for.
No, that's good. I'm going to interject a couple questions, though. Yeah. In your study, are you looking at what is being called at this conference normal patients as well as MS patients?
Yeah, it's called the healthy control group compared to the MS patients group.
And how many are in each pool?
Depends on the study. We have right now to look at the oxygen consumption. That study is like 30 to 30. 30 patients, 30 controls. But the studies published is still under submission. That is only 9 patients and 9 controls to look at the oxygen delivery problems. The matter is called the cerebral vascular reactivity. Right now, we are still collecting data. But I think that's a very interesting, exciting field to go. This is the first, I think, the paper, if we submit and accept it in this field, to really look at the early changes of the vascular hemodynamic changes.
So what method is being used to measure perfusion?
We use, as Dr. Hubbard mentioned, this called arterial spin labeling.
What is that, though? I don't understand what that is.
It is a technique right now. We don't need to do. Usually, using MR, we usually use the inject contrast agent to measure the perfusion or the cerebral blood flow. But this technique, we just label the lower level of the arterial flow, and then to measure the higher level and giving kind of the certain time. Then because we actually got 2 scans. One is labeled scan, one is without labeled. So we kind of use these 2 scans to subtract the signals, because the signal is different. One is saturated, it's called label scan. One is not. So based on these signal changes, we know the blood, how fast the blood goes there and perfuse to the tissue.
So would you say, based on your work, that poor perfusion is a biomarker for MS and other neurovascular diseases?
No, the perfusion imaging, there are several rules. One, mainly, it's a matter of the hemodynamic changes. For example, the cerebral blood flow and cerebral blood volume, the changes. We use this one to measure the cerebral blood flow, to see the flow changes, and the term of the cerebral vascular reactivity is another measure. We use actually the CO2, the carbon dioxide. So we know the carbon dioxide is also the potent the vasodilator. So we ask volunteers or the patients to breathe the room air first, then to breathe the CO2, then to see the difference of the blood flow changes. So that difference tells you how dynamic of your vessels, how good are your vessels, because in normal condition, the vessels dilate quickly and deliver the blood and oxygen quickly. But in MS, we think there's a problem to deliver this efficiently to the site of the neuronal activity. So that's caused neuronal dysfunction.
But she asked, perfusion is low in MS, correct?
Yes.
Yeah.
Perfusion is low. I think we measure usually we measure the local, the regional, the perfusion. I think one of the reasons is, as I mentioned, is not so sensitive, because the vascular smooth muscle is desensitized by the nitric oxide. So that's why, the perfusion generally not so quickly to deliver, because this mechanism, the neurovascular coupling mechanism is disrupted.
But there have been 5 studies using dynamics, susceptibility contrast, and ASL, and earlier PET studies, all of them showing that perfusion by any method is lower in MS than normals.
Yes.
Correct.
Yes.
Yeah.
Yes.
Yeah. But would you not call it a biomarker? She was asking whether that would make it a biomarker. It would be nice if it was, because right now the biomarker for MS is waiting 5 years and counting how many lesions someone has.
Yes.
Right.
Yeah.
So if we had a way to have a way to, it's like blood pressure and stroke.
Yes.
For stroke, you got to wait 5 years and see how many strokes. Blood pressure, you know right away. Would you say that perfusion is, in a sense, a biomarker in that sense, or is it too strong to say?
I think it's a little bit early stage to see that, because a lot of studies are still going on. We didn't get a definite the conclusion from the perfusion studies yet. But I think it's a very good the measure or biomarker, you can call it, for the functional imaging, because as Dr. Hubbard said, the conventional imaging to measure kind of structural changes in lesions and atrophy. These are all structural or kind of late phase changes. But the perfusion can be very early changes, and also the perfusion is dynamic, and it's related to the function of the neuronal activity. So it's more, I think, useful in the future.
Yeah, that would be my hope. Yeah. Right now, we have to do these huge trials because all we have is clinical EDS scores, which are not very objective,
yes.
Lesion counting. So we have to do these very, very long studies. If it turned out that perfusion could give us answers more immediately, we could try medications more quickly. We could try. So what we're doing is we're using it before and after venoplasty, and we have shown that the day after venoplasty, perfusion, ASL perfusion, is a little bit increased. Not a lot, at least to my read. Normal using ASL is normal people have maybe 60 milliliters per 100 grams per minute in the gray matter and 40 to 50 in the white matter. Does those numbers seem in the ballpark? Whereas for us, our MS patients, their gray matter and their white matter are both in the 32 to 35 range. Interestingly, in some cases, the white matter was higher. So we've only done about 10 patients so far, but we have gray matter that's averaging about 33, white matter that's averaging about 35, surprised me. And then after venoplasty, it goes up, but not that much. It goes up to 38, 39. It doesn't go up to 50, 60.
Yeah.
Does that surprise you?
No, actually, I think your results, your study is very interesting. So especially the board study showing the decrease of the board activities in MS patients compared to controls. So that means something was wrong there already. So that's the board I'm talking about is called the blood oxygen level dependent technique. It's measured, when measure the neuronal activities. So. So in Dr. Hubbard, the study, they found a general decrease of the neuronal activity in MS patients. So we found actually similar, the findings. We found also the decreased, this called the cerebral vascular reactivity using board, also the board signals.
And hypercapnia?
Yeah.
Wow.
Yeah, the hypercapnia.
At the same time, you were doing a cognitive task, or?
No, we're doing the board. To ask the subject to breathe the room air for 1 minute and breathe room air for breathe the CO2 for 1 minute. They will repeat 3 times using the board technique. So we measure the ball signal changes.
That's what you presented today?
Yeah, yeah.
You were presenting ball data. I didn't get that.
Yeah, yeah.
That's not a good sign.
Yeah. So the ball, we know the neurovascular coupling. So when the blood flow increase, in this case, it's due to the CO2.
Yeah.
So then they suppose the ball signal increase too because they bring a lot of oxygen. Right?
Yeah.
So we found so the MS the patients group there's this increased signal is not as strong as in controls.
I was thinking, listening to you, could I use caffeine in the same way? Could I collect my data and then have them take a dose of caffeine and then collect it again? Would that
Yes.
tell me something similar?
Yes, yes, yeah.
It's easier than that device to do the
Yeah,
CO2.
it's easier, but probably that's the opposite way because the caffeine is a vasoconstrictor.
Yeah.
It's not a vasodilator.
Yeah.
But you can test the vaso.
Compare the two.
Yeah, compare the two, and also you can test these vaso reactivity things because you just test the tune of the vascular smooth muscle, whether it's healthy or not.
Yeah.
Yeah.
Okay, thank you, gentlemen. That was very interesting. And I appreciate you both helping each other steer the ship. That was good. Frank, would you pause
I was
that?
picking his brain.
Ready, Frank?
Rolling.
Dr. Hubbard, can you tell us a little bit about your national approach to gather data for MS outcomes? Fill us in a little bit on what that is and what you're doing.
When my son was diagnosed, and I arranged to have him have the venoplasty, actually while he was on the table, he said, "Dad, make sure that other patients are going to have access to this too." This was in May 2010. Mike Dake had not had, the IRB had been not allowed. Sal Scafani at SUNY Downstate, he had been turned down by the IRB twice. There were no IRB studies approved in the United States at that point. Canadians were completely shut down. They were not allowed even to be tested, much less treated. So I decided that I needed to find a way to have people be treated, and that meant I had to find a way to have an IRB approved study. So at first I got an IRB approval just in San Diego with 3 interventional radiologists, one of whom was the one who treated my son. It was the first one he had treated. But then that was also very tough because it meant everyone had to come to San Diego. So basically I got IRB approval for a multi-center registry. So a registry by definition means it's not double-blind, it's not placebo-controlled. So and it's quite appropriate to belittle registries because they will not be able to prove whether venoplasty in fact is an effective treatment for MS. But I think it's hugely important for 2 reasons. One of which is you can collect adverse events. If it's true that 15 or 20,000 people around the world have had this treatment, only maybe 1,000 have had their data collected in any way. Our first study had 300 patients. Our registry at this point, I think, has about 500 patients, so that's a total of about 800. And then there's a couple of other small studies. There's Siskin in Albany, there's Buffalo, very small numbers, right? Buffalo, they're doing this double-blind study, but it's 10 or 20 cases. Saskatchewan has said they're going to do a project with Gary Siskin, and that'll only be 88 to 100 patients. So there's a real need for that. So basically we created a multi-center registry. So big drug companies, they don't get IRBs in every single hospital. They go to a national IRB, of which there are a few around the country, where one IRB oversees the credentialing of physicians all across the country. So right now we have 26 sites around the country. Most of them are on the East Coast, but there's one in Seattle, there's one in Houston, Dayton, Ohio, Memphis, Atlanta, San Diego, where there's an IR who is part of the study. His data is collected by us, so he sends us his adverse events data. He sends the venography data, how many balloons he used, size of balloon, what locations he did, and then we have the patient fill out quality of life measures before, 1, 6, and 12 months after. So at this point, 26 sites, I think there's about 300 that are enrolled. So we are collecting that data. It does two things. It gives patients the reassurance that their data is being counted, that there's some measure. It gives everyone some comfort about adverse events. One of the issues right on is poor Mike Dake, you know, fantastic guy. He had a death right early on. Through no fault of his own, but the patient was on anticoagulation and had bleeding. So the people who are against this, in general, the neurology community, I'm a neurologist, but I'm pretty much one of 2 or 3 neurologists on the planet that don't hate this whole thing. They love to focus on how dangerous it is. Well, because of our multi-center registry, I'm able to report officially that there have been no deaths, no serious adverse events, and a very small number of very minor side effects. So we're also going to present our clinical outcomes data. In our case, our first 260 cases, we've been accepted to the Interventional Radiology Journal. It hasn't been published yet, but it's been conditionally accepted for publication. We've shown essentially no side effects, and using the MSIS, which is the Multiple Sclerosis Impact Scale, we've shown that about 60% of patients are having a significant improvement. At the significance level P less than 0.01, so very statistically significant. Now, the neurologist, when this paper comes out, they're going to say it's the worst paper that ever was written because it's not double-blind, placebo-controlled, and that's absolutely true. But still, it's a start. It's a start for the patients to know they're going somewhere where people are looking at them. It's not some guy who's kind of doing it in a warehouse, you know, at night. It's good for the IRs because the IRs can say to their colleagues that doing this under an IRB-approved trial. That's what we're doing.
Would you now consider this to be national? I mean, you talk about these 26 centers and some other, you know, locations that are gathering data, but would you consider this sort of the national registry at this point?
If you went to the website and you looked at the map, Google Map, you can Google Map Hubbard Foundation CCSVI registry. It looks national. There are little red dots all over the country. All the well-known IRs are doing it. There are 2 large groups. One's called Vascular Access Center. The main doc is James McGuckin. He'll be here tomorrow. And the other one used to be called American Access Centers. It was bought out by a big dialysis company called Fresenius. So this is a large group. This organization has all the key people that are doing the procedure. There are apparently other people doing it kind of quietly, not collecting any data. Between this organization and our national registry, all the people who are serious about this are being evaluated.
So will they at some point in time join your registry so that all of the data is in one cohesive place? I mean, will this become sort of a PubMed type of one-stop shop?
It makes sense. I think the politics will get too tricky there. The organization, Society of International Radiology, I would think, my read is they'd want to have their own.
Okay.
So I don't think so, but that's okay. I think one could argue it's better to have a few registries rather than one. Ultimately, though, we're going to have a double-blind placebo-controlled trial. Even if we don't care about getting the neurologist to be interested, insurance companies are not going to pay for it. Mm-hmm. These patients are having to pay out of their own pocket. Typically, they're paying $7,500 for the interventional radiology, and for the Doppler or MRI studies, they're probably averaging about $1,000 or more. So they're having to pay for that out of their own pocket because insurance companies are saying this is not yet proven. In our case, because the neurologists are so against this, I mean everywhere, we require a prescription. We require that the patient have normal kidney function. So that means a physician has to get them a kidney test, and then we require that some physician is writing an order for an MRI. You can't just get an MRI, right? You can't walk into your local mall and get an MRI. Some doctor has to order it. In general, it's being ordered by the patient's family docs, not their neurologists, right? So it is possible that the neurologists are going to just get left out. Neurologists do this. Again, I'm a neurologist. We used to have psychiatry. Now psychiatry is a whole different field, right? It used to be neurologists treated mental illness. That's been gone for 100 years. But we used to have sleep disorders. Now it's pulmonary doctors that have sleep disorders. We used to have dementia. Most dementia patients now are being treated by geriatricians, elderly doctors. Neurology needs to wake up. They're at risk for losing another disease. They're running out of diseases that they can be cavalier about. But the insurance issue to me is key. Unless there's a pivotal trial in the FDA terminology, pivotal trial is a trial on the basis of which the FDA will approve a drug. So I think that's why we have to have a trial. Why are people against it? Number one, who's going to pay for it? That's going to be a very expensive trial. No patient's going to pay out of their own pocket for a sham. And then number two, how are you really going to be able to disguise it? Blowing up a balloon in the chest or the neck, you can feel it. So there are those who say, geez, how's that going to even work? So I'm not sure what the right answer is on that.
But in terms of what you're doing here, won't this indeed in time help you to get that funding that you need to take it to the next level?
Well, so funding. So, right, there's 3-4 sources of funding.
Okay.
There's NIH, there's Big Pharma, and there's private donations, right? So Big Pharma and NIH, they're not close to doing this. They are totally $50 billion invested in the unproven autoimmune hypothesis.
Oh.
Right? They're not going near this for, I'd say, indefinitely, 10 years minimum. So that means private donations. Most private donations go through the MS societies, National MS Society, and there's 3 in the United States, and then there's 1 in Canada, MS Society of Canada, right? Their donations are down by at least 50% because the patients are so mad that the MS societies seem to be ignoring their desire to have this be looked at. The Hubbard Foundation, so my family's foundation, that's what we see our mission as. We see our mission as trying to raise money, private donations, to raise enough money to do these trials.
Okay. What have I not asked you, Dr. Hubbard, about this that you think is really critical to share? If you could share with a private donor, for example, or somebody that's considering a donation and wants to support this, why should they? Why would their dollars here perhaps be more meaningful than to the 20 other causes that they might be being solicited for?
I think after 50 years, we're actually close. 50 years, we've been looking to prove that myelin, the lining of the brain, is being attacked by the immune system. It shouldn't take 50 years and $50 billion. It's over, but everyone's attention's over there. Suddenly there's an idea. I hope you're going to interview Paolo. Paolo's just a beautiful guy. Came up with reminded us about the vascular hypothesis. So there's an amazing window of opportunity. I think that window of opportunity is going to be hit with perfusion. I think perfusion could be the test that shows why CCSVI of the neck is having an effect on the brain, right? I mean, that's what we need to know. So perfusion answers that, and also it answers the basic scientific question that neurologists need to know. So it may not turn out. I was very pleased to have him kind of say he thought I was right about that, because that's quite a leap to decide that perfusion might be the biomarker. But there's a possibility of it, and if so, it would not be that expensive.
Right.
It would be $3-4 million. So to me, there's a nice window of opportunity here.
Thank you so much, Dr. Hubbard.